Last time we were talking about stem morphology, and the whole purpose of most stems is to prop the leaves up so that the leaves can get at the sunlight. The leaves need to get at sunlight because their main function is to photosynthesize and produce sugars, as we've talked about before. So we're going to talk about leaf morphology in this module. And we start with the outermost layers of the leaf. This is known as the leaf epidermis. I'll go ahead and label that. Notes down here. The epidermis is a single layer of cells. You'll find an upper epidermis and also a lower epidermis on a leaf. And its main function is waterproofing. It has a layer of wax that waterproofs the leaf. And when I say waterproofing, it's not to keep water out of the leaf so much as it is to keep water inside of the leaf, or to prevent the plant from losing too much water through its leaves. The epidermis, especially the underside of the leaf, is covered with numerous pores, and the pore itself is called a stomata or a stoma. Stoma is singular for stomata, and the stoma is surrounded by two cells called guard cells. And so let's talk about the function of them. We've got guard cells, which basically open and close the stomata, or the pores of the leaf. They're found mostly on the underside of the leaf. They will say open or close the stomata, which are the pores of the leaf, and they're mostly on the underside of the leaf. Another portion of the leaf is tightly packed columnar shaped cells, and they're found directly below the upper epidermis, and these are called the palisades layer, or the palisades cells. And if you look closely, you can see these little dark spheres or little dark shapes inside of these palisades, and those are actually chloroplasts. And so the palisade cells, their main function is photosynthesis. Or photosynthesizing. In the lower portions, let's put it on this side here, you find more loosely packed cells with various air pockets in them, and those are referred to as the spongy, because they're full of holes or spaces, spongy mesophyll cells. And they do have some, I'll write it again down here, spongy mesophyll. It's also in your notes. They're loosely packed. Once again, there's lots of air spaces. I'll say loosely packed cells. These air spaces in here allow for gas exchange. In other words, it allows them to uptake the gas, specifically carbon dioxide. And so we'll say the CO2 is found in these spaces in here. The CO2 gets in initially through, guess what? The stoma. And these spongy mesophyll cells absorb it. Now, you'll also see that here and also here are some vascular tissue, xylem and phloem, in other words, or we'll just say veins on this leaf. And so this is the typical morphological structure of your generic sort of leaf. In addition to the inner structure of the leaf, leaf shape can also tell us some information about how a plant operates. And in particular, it can tell us whether or not a plant is a monocot or a dicot. And if you recall from our earlier discussion, monocots have one seed leaf. Again, it's a seed leaf. It does not mean the plant only has one leaf. I don't want to see that or hear that back from you. It is a one seed leaf plant. It's called a monocot. Monocot leaf shapes are typically long and strap-like, and as far as the vascular tissue is concerned, the veins run parallel. And so long and strap-like veins running like this says basically to me grass-type plants. And indeed, all the grasses are monocots. And then on the other hand, we've got dicots or dicotyledons, which means they have two seed leaves. And they have more of a network vein pattern in their leaves. In addition to that, the leaf shapes are much more variable. We refer to dicots as broadleaf plants, and here's our branched veins, and these little side veins will have also their own branching. And so we got monocot, grassy type plants, and dicot, or also referred to as broad leaf plants. And so let's look at a couple of actual photos depicting this. And here we have on the left a leaf from an iris monocot. Again, parallel veins. And on the right here we have a dicot leaf, which happens to be from a sycamore tree, and you can see the secondary branch pattern of the veins on those leaves. Now this covers the typical photosynthetic type leaves, but like we have modified stems, we also have modified leaves as well. And they come in a bunch of different forms and shapes. And they include, I'll make a list here, modified leaves. They would include things such as thorns, okay, which are modified leaves. Many plants are equipped with thorns. Various types of plants instead of these typical leaves up here, including plants that we call the gymnosperms, have scale-like leaves, which are very thick and leathery. We refer to those as scales. We can also see tendrils. A tendril is basically a modified leaf that you commonly see on trailing or vining type plants. Many of the beans grow tendrils, where you will have a stem, and instead of a leaf growing out, you have a long filament type appendage here, and it starts to coil eventually and basically hopefully catches on something and allows the plant to climb or to vine upwards. There are various similar structures out there, claws, little suction cup type devices that allow a lot of plants to climb up very, very smooth surfaces. And this would also include, finally here, the modified leaves, what we'll discuss in the next module, which are flowers. Flower parts are all modified leaves, and they grow from the same sorts of buds that these leaves or any of these other modified type leaves would grow from. And the difference is when they start to differentiate, they became flowers, or they became claws or thorns, or whatever the case may be, or photosynthetic leaves. And so until we reach the differentiation stage, all of these start as your basic subapical meristem.